Good morning friends, welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give your valuable comments. You can also visit the playlist. In the playlist we have provided the topics so far we have covered in a very systematic fashion, and this is quite convenient for the study purpose. And you can visit the playlist. In the playlist we have provided different topics related to comorbidity with separate headings, and maybe the PG scholars, maybe the UG scholars, if you follow this, I hope this will be very convenient for you to study in a regular way. Anyway, my dear friends, today onwards we will going to start a new series of talks, and this is regarding common bleeding and as well as coagulation disorders in children. So bleeding disorders and clotting disorders are there, and bleeding and clotting disorders, they also form a big portion of comorbidity. Often we will see in the OPD also different types of bleeding and clotting disorders, especially in the newborn. We have different types of bleeding and clotting disorders, early hemorrhagic disorders, late hemorrhagic disorders of the newborn. Okay, so hemophilia. So these are all different types of the bleeding and clotting disorders which is very commonly seen in case of children. So we shall have a detailed description of this bleeding and clotting disorders in coming classes. Now, first let me understand what do you mean by normal hemostasis? So hemostasis means it is blocking the bleeding. Usually in common terms it is called as hemostasis. Stasis of the flow of the blood from the vessels to outside. So actually when any sort of injury takes place, our body does not want to lose any blood. This is a normal body physiology. As soon as any injury takes place, the body will not want to lose any blood because it is very precious for it. So loss of the blood will be prevented by different types of processes, and these processes by which the loss of the blood will be prevented from the body, and that is by means of formation of a clot. Okay, at the site of the injury there is formation of the clot, and because of that clot the blood flow will be restricted or blocked, and this process is called as hemostasis. This process is called as hemostasis. Now in the normal hemostasis there is involvement of the three important factors. Number one is blood vessels, number two is platelets, and number three is clotting factors. Now what is the role of the blood vessels in prevention of the blood flow from inside to outside after injury? Now after injury there is a reflex vasoconstriction of the vessel at the site of the injury due to vasoconstrictor substances like endothelial cells. Now actually the vessel is lined, the underneath layer of the vessel is called as endothelial cells, endothelial cell layer. Now, as soon as the injury takes place, the endothelial cell layer that will produce some of the very important substances, which are called as vasoconstrictor substances, and as a result there will be vasoconstriction of the blood vessel, and hence the flow of the blood is restricted or prevented, and blood loss will be minimum. Similarly, there is also decrease in the diameter of the vessel at the injury site. Another reflex mechanism which is taking place, as soon as the injury takes place, the diameter of the vessel which is supplying to that particular area, so that will be also get reduced, and that also leads to less loss of the blood. So this is how the blood vessels are playing their role in prevention of the blood loss after injury. Now platelets, now the second important role is from the platelets. Platelets will form a structure at the site of the injury, at that particular gap. What the platelets will do is they form a structure which is called as temporary or primary hemostatic plug, which is called as temporary or primary hemostatic plug. And this is as soon as the injury takes place, the platelets will form this particular structure. This is to prevent the immediate blood loss. But this plug is not a very firm structure. Okay, so this is called as primary hemostatic plug because it is a weak structure. For time being, as an emergency, it blocks the blood flow. But it needs to be solidified further. It needs to be strengthened further. So therefore, this plug is called as primary hemostatic plug or temporary hemostatic plug. But if the flow of the blood is more, this plug can be washed away because this is not a firm plug. So we need to produce a strong clot at that particular site. The plug should be very strong. Otherwise, this temporary plug may wash it away. So to make that plug, temporary plug which is created by or produced by the platelets more stronger, now we need the clotting factors. So this leads to so in the process of strengthening of this primary hemostatic plug, there is stimulation of the clotting factors. So now the coagulation cascade will be activated. Coagulation cascade means there are so many clotting factors are there, 1 to 13. One by one they will go into activate. That activation we will call it as coagulation cascade. Okay. So once these clotting factors are get activated, now they produce the fibrin. Okay. Ultimately they convert the fibrinogen into fibrin, which is a mesh-like structure, and these mesh-like structures come and deposit over this temporary plug formed by the platelets, and that leads to the formation of the permanent plug, which is a firm one. Okay, it cannot be washed away by the blood flow so easily. It's a firm plug, and that is called as permanent plug. So therefore we have primary hemostatic plug or temporary plug followed by the permanent plug. Okay, so formation of the primary hemostatic plug is basically by the platelets. Initially. But strengthening of that particular hemostatic plug, primary hemostatic plug, is the work of the clotting factors. The strengthening is possible only after the activation of the clotting factors. Now this is, if you see the picture of the blood vessel. Suppose if you take, this is the lumen of the blood vessel, the innermost layer is called as endothelial cell layer. Then just above that there is one more layer which is called as subendothelial cell layer, and over that we will have deposition of the collagen substances, and over that we will have the arterial muscular layer, okay, which is also called as tunica interna. So when there is a cut, what happens when there is a cut? See, the vessels are get affected. So vessels undergo vasoconstriction. The diameter of the vessels also get decreased, so that blood flow will be reduced, and then there will be formation of the temporary plug, and this will be followed by the formation of the secondary plug or permanent plug. Now let us look into the factor how the platelets will activation. The first is vessels. We know that it is a reflex action. There will be vasoconstriction of the vessel, and as well as there is a decrease in the diameter of the vessel, so that blood flow is restricted. But more important, to block that particular injury site is the formation of the platelet plug. Now let us see how the platelets are get activated immediately after injury. Now when there is injury to the endothelial cells of the vessel, there will be expression of the Weibel-Pollard body. Okay, so this particular expression by the VP body, so that will going to produce a factor which is called as VWF, that is von Willebrand's factor. Okay, now this is the first step in the platelet activation. Endothelial cell injury takes place, so that will go into, as soon as the injury takes place, there is expression of a substance in the body called as Weibel-Pollard body, and this particular VP body, so that will go into produce a factor which is very, very important factor, which is called as von Willebrand's factor. Now, as soon as this substance is produced, the platelets which are moving the circulation are get attached to the von Willebrand factor through a receptor which is called as GP1B. Okay, so that means platelets are moving themselves in the flowing blood. There is an injury at the vessel in a particular site. Immediately there is production of the von Willebrand factor. Now von Willebrand factor will call the platelets and it is get attached to the GP1B, and as a result so many platelets come together and get adhered, and this is called as the first step in the clotting. So that is called as platelet adhesion. So platelet adhesion is possible only in the presence of the von Willebrand factor. Okay, there is some disease, there is one disease called as von Willebrand disease where this von Willebrand factor is absent genetically. So that will lead to problem in the platelet adhesion in the initial part itself. Okay. Okay, now von Willebrand factor is produced that will cause the platelet adhesion. It will call up so many platelets together, and so many platelets come together and get adhered together. Now, once the platelets are get adhesion, then there is platelet activation. What do you mean by platelet activation? That means platelets, they come together and adhere. Now, activation means there is change in the shape of the platelets. And now they start releasing some of the granules. That means granules which are present inside the platelet, so they now get released into outside. We will call it as degranulation. So we will call it as degranulation. Now, when this degranulation takes place, this causes an expression of a molecule called as glycoprotein 2B. Okay, as soon as the degranulation takes place, this causes expression of a molecule. That means production of a molecule which is called as glycoprotein 2B and as well as fibrinogen. So formation of this glycoprotein 2B and fibrinogen now helps to attach other platelets, many platelets together, and many platelets comes together and they get aggregated, and this is called as platelet aggregation. So now we have got three important steps. One is formation of the von Willebrand factor which cause platelet adhesion. Second one is there is degranulation taking place. Once the degranulation takes place, there is production of glycoprotein 2B and as well as fibrinogen. But once this fibrinogen and glycoprotein 2B are produced, so that will cause adhesion of more and more platelets, and this leads to the platelet aggregation. So platelet adhesion and platelet aggregation. Now this is, if you want to see pictorially, the first is platelet adhesion. Okay, so platelet adhesion, which is also called as primary hemostasis, temporary hemostatic plug. Then there is platelet activation. GP2B molecules are coming, fibrinogen is released. Okay, so then platelet activation takes place, and later it will be followed by the platelet aggregation. Once this fibrinogen and glycoprotein 2 molecules are produced, so they will attract so many other platelets also together. They all come together and get binded together and form a plug-like structure. Okay, so hence there is a formation of the secondary hemostasis. Primary hemostasis is over. Now there is formation of secondary hemostasis, and once the secondary hemostasis occur, now this needs to be get strengthened. For the strengthening of this secondary hemostatic plug, we need primary hemostatic plug. We know that by the platelets, which is very weak. So that will be little strengthened by means of secondary hemostasis. Again, this is also not so strong. This is also weaker one. This needs to be now strengthened more, for which we need the activation of the clotting factors. So talking about temporary hemostatic plug, this platelet aggregation responsible for formation of temporary hemostatic plug, this requires three steps we have already discussed. Okay, adhesion of the platelets, activation of the platelets, and platelet aggregation. Adhesion means glycoprotein 1B and von Willebrand factor. Activation, degranulation process, and also there is production of the thromboxane A2. platelet aggregation means glycogen 2b production and fibrinogen production. But still this is very, very weak. For this, fibrinogen has to convert it into fibrin. Now, how can you make this particular plug a stronger one? The fibrinogen which is present, that has to be get converted into fibrin. Fibrin is a mesh-like substance which causes the stability, which gives the stability to the plug. Okay, so we have to convert this fibrinogen into fibrin. Now, conversion of the fibrinogen into fibrin is only possible by the coagulation cascade. Okay, so once it is converted into fibrin, it becomes very strong plug. Okay, in the whole process now, coagulation factors are very, very important. Only after the activation of the coagulation process, coagulation cascade, this particular fibrinogen can be converted into fibrin, and this fibrin, okay, so silky and mesh-like substance, so that will come and get deposited over this plug, and the plug become more stronger, which is also called as permanent hemostatic plug. Okay, so conversion of this water soluble weak fibrinogen into water insoluble strong fibrin, so that platelet plug is get strengthened and bleeding from the spot stops permanently, is the work of clotting factors. I hope you understand why this fibrinogen has to be converted into fibrin. The reason is fibrinogen is water soluble. It is very weak, so it will going to dissolve if it is water soluble. It has to be converted into water insoluble substances, which can resist the water flow. Okay, because blood also contains the water. Okay, and stronger one. So how this is possible? This is possible only by conversion of fibrinogen into fibrin. Conversion of fibrinogen into fibrin is possible only by means of clotting factors, and that is the main function of the clotting factors also. Okay, so this is the basic thing related to hemostasis. Rest of the things related to the same we will deal in the next class on our concept of the platelet disorders, and what do you mean by platelet disorders, what do you mean by coagulation disorders, that we will discuss one by one in the coming classes. Thank you very much.